For this lecture, we have a special topic on how to use the MarvinSketch. Now, this is not a required software. This whole topic is optional. You don't have to learn how to use it. But this is a pretty useful software that you can draw a lot of molecules, and it will help you to learn the knowledge. So I encourage you to install the software and just play with it. But if you don't use it, it's fine. You can always use handwriting, hand drawing, and if you finish a assignment or the exam without using MarvinSketch, I will not detect any marks from you. If you use MarvinSketch, then you make sure that you do it correctly. I won't give you extra score for using it, but this software will help you make the correct structure. Now, I assume you already have installed the desktop version of MarvinSketch. MarvinSketch is also available online, which is called the MarvinSketch JS, stands for JavaScript version. That one is less powerful than the desktop version, so it has both the Windows version and the Mac version. So yeah, just install the software and try it. And after learning this, you should be able to know the user interface and be able to find different settings of this software. After this lecture, you should be able to draw biomolecules, some of the biomolecules, and draw the hydrogen patterns between molecules. So be able to draw peptide structure based on the amino acid sequence. And you should also be able to draw stereoisomers in a specific configuration. Now, after you install this software and launch the program, you're going to see an interface like this. So this one is version 1925. The latest version is version 20 something. And basically, it's the same. Now, you have on the top the menu bar, which you can click and it will shows up different options. Here you have a generic toolbar that including the selection, erase, undo or redo, and cut paste, select by structure and zoom. On the left, you have these tools, the toolbar. So you can draw a bond or a chain and different conformation or different type of chain. You can also input the text, and there's many other options adding this charge. And at the bottom, you can see this advanced template for drawing chemical structure with 3-membrane, 4-membrane, 5-membrane, etc. And here is the status bar on the bottom, which is similar always to many other softwares. It tells you whether the structure you draw is a 2D version or not, or 3D version. On the right, you have these atoms toolbar. So here you can see the periodical table. You can select different atoms, the most commonly used ones, proton, carbon, nitrogen, oxygen. These are the ones most often used in the biomolecules. So the central area is the canvas where you draw these molecules. Now this is the question of hydrogen bond in first assignment, the second question. It asks you to draw all possible hydrogen bond structures between one water molecule and ethanol molecules. I mean, many of the students did not read the question carefully. It's about a water molecule with different ethanol molecules, and they ask you to insert a picture. So you can either draw by hand or draw use the software. So here's the answer to this question. How do we draw it? And I'm going to show you in the next slide. But the first question you want to ask yourself is how many hydrogen bonds is possible to be formed between a water molecule and the ethanols. We know that the oxygen has a sp3 hybrid orbital, so it has two free electron pairs, and it also forms the two covalent bonds with the proton. So basically, it can form 4 of the hydrogen bonds. 2 of them, it's serving as the donor of the hydrogen bonds, so contribute 1 proton each. The oxygen from the ethanol is serving as the acceptor for that. Now, this oxygen over the water can also serve as the acceptor of the hydrogen bond. So 2 of the ethanol molecules contributes this OH group and form 2 hydrogen bonds with this oxygen of the water. So if you did not get the full mark, then here's the reason. Now, before I use the Marvin software to draw the actual figure that I presented in previous slides, let me tell you the 4 steps that you need to do to draw that picture. The first step is to draw the molecules of the water and the ethanol. So you can just type the name sometimes of that. And the important thing you need to know is that the Marvin has this option to expand a group. So functional group, you have a name. If you expand that functional group, then it will give you the right structure. So if you type H2O and expand that, it will expand to a structure like this. So this is shown here. It's covered, but that's how it works. The second one is that when you draw a ethanol, it will start with a functional group like this. There's no single bond between the OH group, so you need to add a explicit hydrogens. So this is a second trick. The next one is that between the hydrogen bond acceptor and the donor, you need to make a bond. It cannot be a covalent bond like this. So the correct bond you need to add is any bond. So if you have, let's say, a ethanol here, and then you need to make a bond between this proton and this oxygen. Now this one, it's sometimes it's just any. If you want to make it more nice, you can change that bond pattern to a hashed bond. So these are the 4 steps that you need to use to make a structure of 2 molecules making a hydrogen bond. So besides the hydrogen bond, I will also show you how to use the MarvinSketch to convert a peptide sequence into a structure, and also draw the structure that cannot be simply described by the amino acid sequence. For example, the glutathione, you know that it's a tripeptide, and there's isopeptide between the glutamic acid and the cysteine. So this is not a typical alpha peptide bond, and you have to draw this manually. So please watch the several videos, and I'll show you in using the software how to draw the structures.